So today we're going to talk about the nucleus, which is the largest cell organelle. We have discussed chloroplast and mitochondria in detail. And if you look at a nucleus, okay, this is a nucleus, and the nuclear membrane extends something like this. This is what a nuclear membrane actually looks like. Okay? It exists something like this. And there are two types. We call these membranes reticulums. This is also a reticulum, endoplasmic reticulum, and this is also a endoplasmic reticulum. The only difference is that one of them, some of those endoplasmic reticulums have these ribosomes on them too. The one with the ribosomes we call as rough endoplasmic reticulum, and the one without ribosomes are known as smooth endoplasmic reticulum. Can you tell me what is the function of the ribosomes?
Protein synthesis.
No, yeah, protein synthesis. Okay, good answer. Then we have the nucleoplasm, and inside the nucleoplasm, we have the deeply dark structure, which is known as the nucleolus as well. Nucleus is surrounded by two membranes, forming a nuclear envelope. Outer membrane is always filled with the endoplasmic reticulum, like I've drawn over it. And then there's an inner membrane as well that is not having the endoplasmic reticulum. So basically, this RER and SER is the outer membrane of the nucleus, and this is the inner membrane. Are you getting it?
Yeah.
The nuclear envelope has many small pores called the nuclear pores. These allow and control the exchange between the nucleus and the cytoplasm. Examples of substances, for example, जो exit करती हैं, the exiting substances include messenger RNA, transfer RNA, and ribosomes. And the examples of substances that enter the nucleus include proteins, nucleotides, ATP. What is ATP?
Uh, like, like it stores energy. It's just energy.
Good. It has the energy, it's an energy carrier basically. The nucleus also contains chromosomes, which contain DNA. What is DNA?
It contains genes which like determine our characteristics and
Mhm.
yeah.
Good. DNA is organized into functional units called genes, and genes control the activities of the cell and inheritance. Thus, the nucleus controls the overall cell activities as well. The DNA molecules are so long that they have to be folded up into a more compact shape to prevent the strands becoming tangled. This is achieved by combining with proteins, particularly with proteins known as histones, and the combination of DNA and the proteins is known as chromatin. Chromatin also contains some RNA, and we say that chromosomes are made up of chromatin. The difference only is that chromatin are very clear when they are dense. This is what a chromatin looks like. And when it becomes coiled, we call these chromosomes. And we'll be doing this in detail in your chapter number 6. Chapter 6, this would be a very, very detailed chapter. And when a cell is about to divide, cell division will be studied in chapter number 5. During the cell division, the first nucleus to divide, the first organelle to divide is actually the nucleus. Also, within the nucleolus, a structure called the nucleolus as well. Nucleolus is the most darkly stained round structure in the nucleus, and as mentioned earlier, it is the most common one as well because it's in there every cell. It's easily seen even using a light microscope as well. It can be easily seen as well. It contains a core of DNA as well. It also provides the guidance or the code to produce ribosomal RNA, or what we can call as RNA as well. Where is this ribosomal RNA needed? Where is this tRNA needed? This is also made by the nucleolus. We will study this in chapter number 6. tRNA stands for transfer RNA. rRNA stands for ribosomal RNA. And around the core are less dense regions where the ribosomal subunits are assembled, combining the rRNA with ribosomal proteins imported from the cytoplasm. So the different parts of the nucleolus only come together during the manufacture of ribosomes, which is one of the function of the nucleus. And the nucleolus is the one that controls the nucleus in making its other. So nucleolus is like even the head office, even the boss of the whole nucleus, because it's having all of the criteria, the the making, the formula, and everything that the nucleus has to actually create. So can you tell me what is the difference between the two types of endoplasmic reticulum?
Rough endoplasmic reticulum has ribosomes and then the smooth one doesn't.
Exactly. When electron microscopes came, biologists were actually amazed to see a detailed structure known as this endoplasmic reticulum that was kind of a flattened compartments, or you can call them sacs, that are originating from a membranous. Okay, we call these this shape. ये जो shape होती है, this shape is known as cisternae, and a lot of these is known as cristae. A lot of these is called cristae, and otherwise we call it cisternae. Cisternae is the shape of these membranes. So molecules, particularly proteins, can also be transported through these endoplasmic reticulum and can be separated from the cytoplasm. Now, proteins also travel inside these, and in chapter number 6, you'll understand that protein synthesis actually takes place on the rough endoplasmic reticulum's ribosomes. The ER is a continuous with outer membrane of the nuclear envelope, and there are two types of ER, rough ER and smooth ER. RER is also called because it is covered with many tiny organelles called the ribosomes, and they are just visible as black dots in figure 1.23 and 2.4. And ribosomes are the sites for protein synthesis, and they can also be found free in the cytoplasm. Some of them are on the RER, and some are also swimming around the cytoplasm freely. Smooth endoplasmic reticulum has a smooth appearance. Why? It has no ribosomes. It has a completely different function. SER is responsible for making lipids and steroids. This is the what? The function of SER. How these lipids and steroids are made, this is not in our syllabus. You should just know what is the function of SER. For example, producing cholesterol, lipids, fats, triglycerides, some fatty hormones as well, which is estrogen, testosterone, the sexual hormones, and it is also major storage site for calcium ions. So what do you have to know? What is smooth endoplasmic reticulum? Function is producing lipids, steroids, many other hormones such as the sexual hormones. Then it's also a storage site for calcium ions. And they are most in the concentration inside muscle cells. Why are they present so much in the muscle cells? Because calcium ions are involved in muscle contraction. And how are they involved? You'll study in your chapter number 15 of A2 biology. Got it?
Yeah.
What are ribosomes? The sites for protein synthesis, and where are they found?
RER
And where is the RER?
attached on the membrane of the nucleus.
Ribosomes not always on RER. They some of them are also on the cytoplasm. And when they have to be used, they come and join with the RER as well. Ribosomes are very small and not visible with a light microscope. At very high magnifications using an electron microscope, they can't be seen to consist of two subunits, a large and a small subunit. The sizes of the structures of the small are often quoted in S units. S units are a measure of how rapidly sediment in high speed centrifuge. S is a unit that actually tells you the production speed, something how fast is something being produced. Now there are two types and two sizes of ribosomes as well, ATS and 70S. ATS ribosomes are present in eukaryotes. Can you give me any examples of eukaryotes that you've studied in IGCSE?
Uh, Human.
Yeah, human, animal, plants, good, fungi. And 70S ribosomes are a smaller ones. They are present in prokaryotes. Give me an example of prokaryote.
Bacteria.
Good job, good job. Plus, these 70S ribosomes are a bit smaller and hence they are also present in these organelles. Now you must be thinking that if ATS are present in eukaryotes, eukaryote cells contain ribosome, but eukaryote cells organelles contain what? 70S. Is it clear? It's a bit confusing. Got it?
So the eukaryotes have ATS, but their cells have 70S.
Their organelles have 70S.
Okay.
Okay, for example, nucleus, mitochondria, chloroplast, they have 70S. And prokaryotes, everything has 70S. Because 70S ribosomes are slightly smaller. Mitochondria and chloroplast also contain 70S ribosome, revealing their prokaryotic origins. That is why we see that these two organelles, the mitochondria and chloroplast. They were both originated from prokaryotes basically, and then they became a part of eukaryotes as well. Ribosomes are made up of ribosomal RNA, rRNA, and protein. Their three-dimensional structure has now been worked out, and ribosomes allow all the interacting molecules involved in protein synthesis, such as we have mRNA, tRNA, amino acids, regulatory proteins, together in one place. Next, we have Golgi apparatus. Ever heard of this organelle before?
No.
These are also some membranous flattened sacs which are present in the cytoplasm, something like this. This is the site, and also some vesicles also break off from this side as well, and vesicles are formed from this side. So they keep breaking off from this side. We call them Golgi vesicles. And this whole combined is known as Golgi apparatus. This part is known as Golgi body, and the vesicles are known as Golgi vesicles. The function of Golgi apparatus is modification of protein. Once the protein is made in RER, proteins were made on RER in ribosomes. Now the rough endoplasmic reticulum, you have seen these sites. Okay, let's say a ribosome was over here. Once the protein is made, the protein is transferred along the rough endoplasmic reticulum to the Golgi apparatus. Golgi apparatus modifies the protein into different types. Now you don't know what the protein structure is: primary, secondary, tertiary, quaternary. Okay, and there's also types of proteins that you'll study in the next chapter, biological molecules. Golgi apparatus is responsible for what? Transport and modification of protein. Golgi apparatus, once it modifies the protein produced by the rough endoplasmic reticulum ribosomes, it sends off the protein in its desired location by transporting vesicles. Got it?
Yeah.
Golgi apparatus is a stack of flattened sacs called cisternae. Again, these structures are also known as cisternae. More than one Golgi apparatus may be present in a cell as well, and the stack is constantly being formed at one end from vesicles which bud off from the ER and are broken down again at the other end to form Golgi vesicles. The stack of sacs together with the associated vesicles is referred to as the Golgi apparatus or Golgi complex. The Golgi apparatus collects and processes molecules, particularly proteins, from the RAR. It contains hundreds of enzymes for this purpose, and after processing, it transports as well. So, processing, modification, and transporting, these are the two main functions that the Golgi apparatus actually does. And transportation is done by the Golgi vesicles that actually bud off from the apparatus body as well. Releasing molecules from the cell is called secretion, and the pathway followed by the molecules is called secretory pathway. There are some examples of the function of Golgi apparatus as well. For example, now some Golgi vesicles are also used to make lysosomes, which we are going to study the next organelle, what is lysosomes. Moreover, sugars are also added to proteins. A protein, but now the protein has to be modified. So inside the Golgi vesicles, we can also add sugars to proteins to produce glycoproteins. We can also add lipids and sugars together to make glycolipids. We can also use these Golgi enzymes because Golgi apparatus also provides us with many different enzymes. Because can you tell me what is enzyme made up of?
Protein.
Exactly. Enzymes are also made up of protein. So they are also present in the Golgi apparatus. And for example, when you have to make new cell walls, there are many enzymes needed for the production of the cellulose cell wall. So these Golgi apparatus are usually found very near to the cell wall. Why? So they can easily transport the vesicles needed, the vesicle enzymes needed to produce the cell wall. Plus, they also produce goblet cells. Okay, ever heard of the term goblet cells in IGCSE? What was their function?
I think secrete mucus.
Yeah, exactly, to secrete mucus. Why? What was the function of that mucus?
To trap the dust particles in the throat and like beat so that the cilia will beat the mucus up.
Good, good. So mucus is made up of mucin, and this mucin is basically secreted by Golgi apparatus. Got it?
Yeah.
So function one, processing and modification of protein. Function two, transportation of modified protein. Number 3, production of lysosomes. Number 4, making glycoproteins. Number 5, making glycolipids. What are glycoproteins, glycolipids? Chapter number 4. Cell membrane. They are all part of the cell membrane. Then Golgi enzymes and mucin protein, which is needed for the mucus inside the goblet cells. Clear?
Yeah.
Good. I know these are a lot of new terms, but you have to revise daily. Okay? Then you'll be easy. Okay? And I'll be giving you some short notes to write down as well so we remember it in class. Now, lysosomes were produced by Golgi apparatus. What are these? Simple vesicles. This is lysosome. This is a lysosome. These are all lysosomes present in a cell, and they have 0.1 to 0.5 micrometer diameter. They are also in plants as the central large vacuole may also act as a lysosome. These lysosomes are just vesicles. In plant cells, it's one large vesicle. In animal cells, there are numerous small vesicles, and they are present where? In cytoplasm. Lysosomes' function is that they contain digestive hydrolytic enzymes, which also perform the function of destroying old cell organelles. Now, any worn out organelle, such as an old mitochondria, an old piece of Golgi apparatus, it actually eats up that old worn out organelle. And what does it do? Now, look at this. This was a lysosome. It went to digest a worn out organelle. Then the next step was inside the cell. Now the lysosome has digested that worn out organelle. Now this lysosome is going to go towards the cell membrane to secrete out that worn out cell organelle. Got it?
Yeah.
This
So
was
it
the
like
lysosome. Okay?
it gets it gets rid of like cell.
Organelles, the worn out organelles inside the cell. Old organelle,
Oh,
old mitochondria, old RER, old ribosome. Got it?
okay.
How?
Yeah.
Because these lysosomes contain digestive hydrolytic enzymes, which break down these worn out molecules. It eats it up. Now, the worn out organelle is inside the stomach of the lysosome. Then this lysosome moves towards the cell membrane and releases the worn out molecule. Clear? Now, let's read it out. Lysosomes contain digestive enzymes, and the enzymes are called hydrolases. Can you tell me what is the meaning of hydrolysis?
I'm not sure.
Think. Hydro means hydra is what?
Water.
Lysis, breakdown. Breakdown by the help of water. Remember that it's a very important term. It's going to be used in a lot of chapters. And the reverse would be condensation. Remember something? Adding water to break down something is hydrolysis. I'll write it over here as well because it is something that will be there. Now, if you have a molecule A, if you want to break it down into B and C, you'll be adding water. This is known as hydrolysis. Now, if you want to make one whole molecule again, you have to remove water. And the removal of water is known as condensation. Is it clear?
Yeah.
You can write it down with you as well. Hydrolysis is the addition of water to break down molecules. Condensation is the removal of water to join molecules. Now, these enzymes that are present in lysosomes, they are known as hydrolases. Lysosomes are responsible for the breakdown digestion of unwanted substances and structures such as old organelles or sometimes even the whole cells. Now, in many cases, you'll study in your chapter number 11 with me, the very last chapter of AS, that white blood cells. Okay, white blood cells also contain a lot of lysosomes. Think, guess, and tell me what is the function of white blood cells and why do you think they contain the most number of lysosomes?
White blood cells help in immunity, right? Like so they have lysosomes to uh remove infected organelle.
Good, good. What they do is engulf the bacteria. In IGCSE, you studied a process known as phagocytosis. Can you explain me what was that?
So like it would like engulf the like pathogen and then it'll digest it. Like, but first the like something like the other white blood cell will bind it and then it stops moving and then the phagocytes like engulf it and digest it.
Good. Now, those phagocytes, as you just told me, have just said it, phagocytes digested. How? Lysosomes. Got it? Those phagocytes had what? Lysosomes. Is it clear?
Yeah.
Hydrolysis works fastest in an acid environment, so the contents of lysosomes are also acidic of a pH around 4 to 5. No need to remember this. Among the 60 plus enzymes contained in lysosomes, some of them are proteases, the lysosomes used to break down proteinous structures, lipases to break down the structures made up of lipids, nucleases to break down nucleic acids. So lysosomes have different types of hydrolytic enzymes depending on what are they going to break. The enzymes are synthesized on the RER and then delivered to lysosomes via the Golgi apparatus. Now imagine enzymes are what? Protein, right? Protein was made by ribosomes, which were on the RER. Once it was made, the the protein was transferred to Golgi apparatus. Golgi apparatus turned that protein into enzyme and added it inside the lysosome. I'll repeat, and then you're going to explain me all the structures combined in this example that what we have studied till now. Enzymes are a type of protein. Proteins are synthesized on ribosomes. Ribosomes are present on rough endoplasmic reticulum. Rough endoplasmic reticulum sent that protein to Golgi apparatus, where the protein was modified into an enzyme, and through the Golgi vesicles, it was sent to produce lysosomes. And now these lysosomes finally contain those hydrolytic enzymes. Now you explain me all these three structures in this same example again. You can do it.
Which are proteins produced in ribosomes,
Hmm.
which are found on the RER. And then the proteins produced move to the Golgi apparatus so that they can be modified. And then uh then it forms enzymes, and then those enzymes form lysosomes.
Good. Now explain me the function of lysosomes.
Function of lysosomes is it contains those enzymes that can like break down old organelles or like that they don't need anymore, like the cell doesn't need anymore.
Or even some of the pathogens that are unwanted by the body. Now, the
Okay.
activities of lysosomes can be split into four categories. The first one is the one you have just told me, getting rid of unwanted cell components. And how do they do? You just use the term of engulfing. Lysosomes can engulf and destroy unwanted cell components such as molecules or organelles that are located inside the cell. The second process is endocytosis, which is kind of a, you can say, engulfing. What does engulfing mean?
Like developing.
Exactly. Taking it, enveloping, a better word. Yeah, enveloping is a good word. Enveloping it. That is known as endocytosis. Movement of substances across membranes, material may be taken into the cell by endocytosis. Taking material in. ठीक है? If a cell wants to take a material in, the lysosomes are going to move towards the cell membrane. And once the lysosome reach the cell membrane, they are going to take in that substance and then return back to its position. Got it?
Yeah.
Then what would be exocytosis? Exo.
Leaves the cell.
Not lysosome doesn't leave the cell. It fuses with the
No.
membrane and releases the unwanted substance. Got it?
Oh, okay.
Yeah, good job. Self-digestion is also one of the functions of lysosomes. They are sometimes also released inside the cytoplasm. Why? The whole cell being digested, which is known as autolysis. Let's call it self-destruction. If the cell on whole has to destroy itself, lysosome is the one that's gonna do it. Clear? For example, after death of an individual, membranes lose their partial permeability. Now, after someone's death, the cells are destroyed. How? Cell destruction by lysosomes, or even the baby cells that are growing into the adult cells. The baby cells organelles are all destroyed by lysosomes as well during pregnancy once they have become normal size, and also pregnant women's cells that make up the uterus. Because what is the difference that occurs in the uterus size? It becomes bigger, it becomes thicker. There's a thicker wall of cells, right? So once the birth has taken place, can you tell me what would happen to that wall of uterus now?
Or it's like it like falls and menstruation. Like
Exactly. So how is that caused? Lysosomes. Because the wall is made up of what? The cells, endothelial cells. You have studied that as well, right? The lining itself. The lining breaks down. And who is responsible for the lining breakdown? The cells, lysosomes. Got it?
yeah.
You remember the examples that I give you? I don't think menstruation over here is the example. When a uterus is restored to its normal size after pregnancy, that is also a good example. Good. Okay. Okay. So let me check if we have done the basics of chloroplast and mitochondria above. So till now in this chapter, let's see what we have totally covered. We have done a lot. Cells are the basic unit of life. Why cells are needed. Microscope is not that important. Now, if I tell you according to paper point of view, you only have to remember the difference between light and electron microscope. Got it? Nothing else. No labeling, nothing. Also, you should also know the difference between plants and animal cells that you already know as well. The basic structure of plant cell, the basic structure of an animal cell, the basics of these organelles, because we haven't started the detail of some of these. We haven't done the detail of mitochondria. Yes, we have covered Golgi apparatus, centrioles and cell walls, plasma. We have to have the detail as well. We have done this basic. After this, this formula is very important. Understood it? Is it clear?
Yeah.
This is asked in your paper number 3. Okay, so what have you thought of paper number 3? Have the school told you something? Have you got a timetable? How many times you have a lab of bio?
I didn't get anything yet.
I guess it's your lab is gonna happen once a week. And whenever it happens, they're going to give you a worksheet. And you have to share me that worksheet as well so we can prepare for your paper number 3 as well in class. Got it?
Oh, okay.
Yeah, I guess either they're going to give you the whole book form at once, or they're going to give you one past paper every week. And I have all the demonstration videos and you'll be performing it as well in live. I'll be solving the questions with you, all of the P3 ones. This formula is important. Electron microscopy, this is not that important. Okay? You can skip this chapter. The electron microscopy, transmission and scanning, very important. The difference between these two types of electron microscope. Difference between light and electron and difference between transmission electron and scanning electron is important. Then the different structures, the labeling of animal cell, labeling of plant cell, microvilli, no need, nucleus, endoplasmic reticulum. We have covered all of this today. Next, we have mitochondria. Yeah. Mm, okay, let's start with mitochondria as well. So mitochondria is known as the powerhouse of the cell. Why?
Because it produces like it's a site of aerobic respiration, so it produces energy.
Good. What's the equation for aerobic respiration?
Uh,
What's the equation for photosynthesis?
Carbon dioxide and water in the presence of
Yeah.
sunlight and chlorophyll is oxygen and glucose.
Reverse it. What does it become?
What?
Plus, now read it backwards. The photosynthesis
Oh, yeah.
equation. Tell me.
Glucose and oxygen becomes carbon dioxide and water.
That is what respiration equation. It's a trick. Got it? That is
Okay.
exactly aerobic respiration. Why is aerobic respiration known as aerobic respiration? Because it uses oxygen. There was an anaerobic respiration as well that you haven't studied, and now we'll be doing this in AS as well. Now, the structure of mitochondria, as seen with an electron microscope, it is also visible with a light microscope, but electron microscope gives a detailed structure of the mitochondria as well. It is something like this: folded membranous structures. The first membrane, the second membrane. And between these first and second membrane, this space is known as intramembranous space. The same is with the mitochondria as well, the chloroplast. If this is the chloroplast, this is also known as the intramembranous space. Intramembranous space means the space between the first and the inner membrane. Their size is 1 micrometer in diameter, important for MCQs. They can be in various shape, sausage shape, round shape, folded shape, and they project into the interior of the mitochondria. Now, these folded membrane are known as cristae. The inner cytoplasm space of the mitochondria is known as the matrix. Got it? So we have the outer membrane, the inner membrane. Between these membranes, we have the intramembranous space. These folded inner membrane is known as cristae, and the cytoplasm of mitochondria is known as matrix. Is the structure clear?
Yeah.
Good. The number of mitochondria in the cell is variable. How can you tell me, would the number of mitochondria differentiate between a muscle cell and a normal skin cell? What do you think?
Muscles have more mitochondria because they need more energy for contraction.
Good job. So that is the reason for the variability of the number. As they're responsible for aerobic respiration, it is not surprising that the cells with a high demand for energy, such as liver and muscle cells, contain large numbers of mitochondria. A liver cell may also contain as much as 2000 mitochondria, and if you exercise regularly, your muscles will start producing this organelle more in number as well. Now, the main function of mitochondria is to carry out aerobic respiration. Although they do not have functions such as synthesis of lipids, during respiration, a series of reactions take place in which energy is released. And that energy is transferred in the form of molecule known as adenosine triphosphate, which is known as the energy carrying molecule in living cells. The reactions of respiration take place in the solution of matrix and inner membrane, which you'll study in A2 as well. A2 first chapter. Chapter number 12 is your A2 first chapter, in which we discuss that aerobic respiration has 4 steps. Out of those 4 steps, one of them takes place in the matrix, 3 of them takes place in the inner membranous space as well. Matrix contains some enzymes as well, and which are also used in the Krebs cycle. Again, no detail needed. It will be studied in A2 chapter. Once made through respiration, ATP leaves the mitochondria, and it's obviously going to spread rapidly to all parts of the cell where energy is actually needed. And how is energy made from ATP? Wherever energy is needed, ATP breaks down into ADP plus a phosphate. This is triphosphate, this is diphosphate, and a phosphate. So whenever somewhere energy is needed, let's say ATP was produced by mitochondria, it traveled to the muscle cells. In the muscle cells, this reaction took place, which gave out energy. Got it?
Wait, so the reaction of ATP to become ADP and P gives off energy?
This releases energy because everybody asks that how is ATP providing us with energy? Whichever cells needs the energy, ATP goes in that cell and breaks down into ADP and phosphorus through the process hydrolysis. This breaking down releases energy. Imagine you are breaking
Okay,
and then it's providing you with energy.
okay.
Yep. And that is all for biology today. Now we're going to write some notes because it's going to become too much for a day, and we're going to revise some things as well. Take a 2 minutes break. Join again with the second link of the class. Got it?
Okay.
2 minutes only.